2 hours. That's great.
All right, welcome everybody. Hopefully you had a great Nobel conference. I know it's been a week since we last met. Let's see, what announcements do I have? Content checks, I am in the midst of passing them back, so if you haven't gotten them back from me yet, I will get them to you at some point today. Nobel conference, did we have a favorite speaker?
It's got to be Carolyn Bertuzzi, right?
Yes, I think she did a fantastic job. She, I think, is an excellent example of a person who can do high caliber research and communicate effectively, which is sometimes really hard to do. Sometimes you get scientists that are super smart, but it's harder to communicate to a general audience. So if you still have to do that Nobel assignment and you need to watch another presentation and you haven't watched hers, I would recommend watching her presentation on glycan. That Nobel assignment for this class isn't due until tomorrow. I gave you a little extra time, simply because I had to watch that video for today, and the scientific literacy assignment was due today, so I wanted to give you a little bit of a buffer. Anything else about Nobel? Fun thing that you did, something you enjoyed?
I really liked the pastry chef's lecture. I think that was amazing. Like, a lot of people forget that the point of having sugar is to have fun. If you're not having fun, there's no point in having it.
Yeah, I did enjoy hers as well. I like how she was able to combine like some of the science and like psychology of sugar into like artwork almost. It was really interesting how she was able to do that. Yeah. All right, well, next year's Nobel is the mighty Mississippi. If you have any suggestions for what you would like to see at a Nobel conference, let me know. I'm on the planning committee for the next Nobel, so I would love to hear a student perspective. What would you like to see for Nobel? Right? Speakers have already been designated, but what other events would you like to see? All right. A couple more announcements. Content check. All right, I know it's been a week now. They are graded, and one of the most common things I saw, understandably so, was how the heck am I supposed to remember all this for my exam, which is a week from today. So I added an extra folder on Moodle of some just optional, additional like worksheets or problems that you could work through. I know a common thing was it's so spread out, and we get this condensed into like 1 or 2 pages. I found some infographics on cellular respiration that I have thrown in here. That's like a 1-page graphic of the big picture. Eventually I'll throw in some information about photosynthesis in this folder as well. So this might be a really useful folder to help you study and organize your thoughts, especially because you're really being tested on like 3 processes. Right? Cellular respiration, I should say 4 because we have enzymes in there. Enzymes, cellular respiration, fermentation and anaerobic respiration, and then photosynthesis. All right. I also noticed on the content check, somebody emailed me and said, hey, I got this right, but it was graded wrong. Yes, that happened. So this is the first year of me using Moodle for these content checks. There are bound to be some hiccups. So what I noticed is for a lot of you, you might have put down the correct answers, but it was in the wrong order, and then it marked it incorrect, right? And I didn't care about order, so I went back through and manually graded all of these to make sure that it was properly graded. So if you notice that, feel free to let me know, because I go through this and I take these quizzes before I make them public to make sure there are no hiccups like this. The problem is I must always put it in the same order as my answers, so I do not see this. So if anything like that ever happens again, email me and let me know. I don't know unless somebody says something. All right. Any questions, concerns, things we need to talk about because it's been a week? before we continue, I will say today is one of my favorite lecture periods because we get to talk about some case studies, and I think it's a lot of fun to think about. So I had you watch that lecture on fermentation, anaerobic respiration, and so I want to make sure we understand the difference between the two. So we're going to start with a review question, right? Why is it incorrect to say fermentation and anaerobic respiration are the same thing? And then if you get finished early, look up some organisms that use anaerobic respiration. They're typically pretty interesting organisms. I'm going to continue handing back content checks.
That's what I was gonna say. So get someone
It's like
Okay, we might be a little lost in our first case study, but we'll take it out. Okay, there you go. I probably have something to hand that to you since I'm standing here. Oh, yeah.
I saw that. Yes. What is that?
It's most probably.
It's almost like you didn't get it. Yes.
I don't know.
Yeah.
I just lost.
It's almost not even.
Yeah. The dentist eyeshadow, she's like overheated. Do as I say, not as I say. Yeah, but it's
I think that was the most productive.
alcoholic. I can feel that.
I feel like I always say pretty good.
Okay, wait, it's blanking. I don't know. I feel like my last one. Is that it? The O, right?
I want there to be another A. You just felt the thought, so just replace it with I should do six.
No, I feel like there's too much. I can feel Jane first. I don't know. You brought it up.
God, that type of
The carrot guy is.
what kind of muffins do you get? They're like,
Oh, he's kind of miserable.
I have to say, I'm a guy.
Why is he like apple? I wanted to like the Big Hill Farm has not that big of a hill, not that big of a It'd be small.
No, we are.
Yeah, I know.
What is it? The farm on
Yeah, I could eat farm on hills.
Well, that's fermented.
Carrot farm, you say.
When you cook farm,
Or it could be like mountain.
Except for
Curve. Big curve farm.
fermentation
Yeah, I was gonna say shade.
relies on glycogen.
All right, so fermentation, anaerobic respiration, they are not the same thing. Why is it incorrect to say that they are? What's different between them? What's one difference?
Respiration, something other than oxygen is used in the last step.
Yeah, anaerobic respiration can still go through all 4 stages of respiration. It just uses something other than oxygen. Right? What about fermentation? What stages does fermentation go through?
Glycolysis and the regeneration of NAD+.
Yeah. So fermentation just goes through glycolysis, but then there's that extra stage to regenerate NAD+, because we don't want to build up of NADHs, because that can be really bad for our cells. It can promote free radicals. It can be very damaging, right? So we need to regenerate our NAD+. What's another difference between the two?
The amount of ATP produced.
Yeah, the amount of ATP produced. Which one produces more? I hear like mumbling. I don't know. I have a lot of energy this morning, and I feel like this class has low energy, so I'm not jiving as well. But I hear the mumbles. Anaerobic respiration, yes, produces more ATP than just fermentation alone. Does it produce as much ATP as an oxygen was the final electron acceptor? Not as much, but still much more than fermentation alone. Is there any other difference that we could note between the two? Yeah?
Like buildup of lactic acids as a result of fermentation.
Yeah, there could be buildup of products like lactic acid or alcohols as a result of fermentation, which we don't see in anaerobic respiration. No. Any questions about these two? So remember, big picture, anaerobic respiration goes through all 4 stages. We just use something other than oxygen as the final electron acceptor. Whereas fermentation only goes through glycolysis and then stops, but has to regenerate the NAD+. Right, so fermentation produces much less ATP than anaerobic respiration and aerobic respiration. All right, if there's no questions, we're going to move on to our first case study. So I'm going to read this whole thing out so that everybody hears it here. And then we're going to answer some questions about this case study. And this is a real study. This actually happened. All right, so we have a 61-year-old man with a history of home brewing. He stumbled into a Texas emergency room complaining of dizziness. Nurses ran a breathalyzer test, and sure enough, the man's blood alcohol concentration was a whopping 0.37%. or almost 5 times the legal limit for driving in Texas. There was just one hitch, right? This man said that he had not touched a drop of alcohol that day. He would just get drunk out of the blue on a Sunday morning after being at church, or really just any time, says Barbara Cordell, the Dean of Nursing at Panola College in Carthage, Texas. His wife was so dismayed about it that she even bought a breathalyzer. Other medical professionals chalked this man's problem up to closet drinking, but Cordell and Dr. Justin McCarthy, a gastroenterologist in Lubbock, wanted to figure out what was really going on. So the team searched the man's belongings, right, for liquor, and then isolated him in a hospital room for 24 hours. Throughout the day, he ate carbohydrate-rich foods, and the doctors periodically checked his blood for alcohol. At one point, it rose to 12%. All right, so somehow he's still getting drunk. I want you to answer these questions. What is going on? Think about what we learned in that lecture that I had to watch before class.
Eat carbohydrates, it converts into alcohol.
Why does it do that? What is it?
Doctor shows all the time.
Yeah. It's a real syndrome.
I watch them as like background noise, so I wound up watching a lot of stuff. I watch mine as background noise.
Sorry, we're focusing.
You've probably heard of our next case study too, the cyanide murders.
If you describe it, maybe. I don't know.
I wouldn't be surprised if you didn't, if that's what you kind of listen to.
I don't even know.
I don't know.
I'd say guys, some bacteria that has a lot of higher
I said alcohol for
yeah. And it uses a lot of
It probably, let me Google it up. I didn't watch the video. I've been sick for a while.
See, I watched the video and I was
Yeah, at some point, I would get lost with this one.
Oh, okay, I see. When using aerobic, the third one is they can use aerobic respiration when oxygen is available. They only use anaerobic methods when oxygen is not available. So they can do both, but I think it's anaerobic. Because I mean, if you typically think of brewing, making alcohol, never make wine, you seal it.
Is it a kidney?
Because it's small.
Isn't it yeast or bacteria?
It doesn't need oxygen.
Oh, because that's like the thing.
Yeah. No, but it produces little, like, it produces like I think they use for every movie. So they're just, yeah, I think if they can just survive on that, then there's maybe they have a lot of like alcohol. They live in glucose buildup, and they do you just know this stuff, or do you consciously No, I'm reasoning about I don't know if I'm right, but let's look it up.
I believe you.
Okay, you always know the answer to everything. Okay, here's the deal. About that, I've been a tutor before. No, I've been a tutor since I was like 12. I tutored English. So, as you can, the point is, I'm good at looking. I didn't know what I was doing. Let me search it up. Google Gemini.
A lot of them it's fermenting other carbohydrates.
Because that's like part of the cycle. You're regenerating.
Yeah, no, that is not helpful. I mean, how are they living on?
Yeah, I think that's
Ha! High substrate consumption to compensate for the low ATP yield limitation of our organization. Yes!
Why don't you cut it? I say that a bunch of times.
I only heard it once.
Oh my god, I say it so fast. An organism.
Maybe I call it on the floor. And you shit. That's why it's How many hours did you use?
All right. right, we have this case study here. This guy is getting drunk out of nowhere, and we're trying to figure out why. What is going on? What did we come up with? Does anybody have a theory? Yeah, this is a real thing. It's called auto-brewery syndrome. What's happening? Carbohydrates are fermenting. If they're going through fermentation, specifically alcohol fermentation, so that alcohol is entering this man's bloodstream. How is this possible? Is the man doing it? Is there another organism involved?
It's probably just gut bacteria.
Likely gut bacteria. And what is his hobby? He likes to home brew. Yeah, he likes to brew his own beer. And in order to brew beer, you need yeast. It's likely somehow he got a large enough quantity, stomach acid didn't kill it, for whatever reason, it established in his gut. Right? And now these yeast organisms are likely going through alcohol fermentation. Right? So when this man eats carbohydrates, it's basically feeding these yeast, giving it its food source to produce more alcohol. All right? How is this organism able to survive on such low amounts of ATP? How is this yeast?
Because they're submerging themselves in glucose. Even though they're only producing 2 ATP per glucose molecule, say they got like an infinity amount of glucose, then they can still survive.
Yeah, they have a high intake of glucose. They're surrounded by these carbohydrates. What else?
They don't need a lot of energy to live.
Yeah, and they're simply smaller organisms, right? They're a single-celled organism. They don't need a lot of energy, right? Compared to us, we are very much multicellular organisms. Okay. Any questions about this? Like I said, this is a real syndrome. It's called auto-brewery syndrome. It is treatable, all right? So if the individual has this syndrome, they can take an antibiotic to help kill off the bacteria that is going through alcohol fermentation, right? Because getting drunk out of nowhere is not good. Okay, it's not safe. It's not good for your health. There's many things wrong with it. So we want to make sure that we have a way to treat it, and it is treated. All right, so this was a real case in Texas. Yeah?
I'm wondering though, if he got this yeast in his body through his hobby, is that just because I've heard that for some people who like to make their own alcohol, that it's not safe to drink your own alcohol, because there might be byproducts that are made, or something like that, you can poison yourself with your own alcohol. Is that why he got this syndrome, or no?
I don't know. Probably not necessarily. I don't know much about home brewing, but I know there are risks involved with making your own types of drinks and alcohol. It could go wrong, it could taste bad, maybe something is introduced that shouldn't be. I'm thinking likely, for whatever reason, that yeast was just able to bypass the acidic stomach, not get killed and established. Maybe he has a genetic predisposition, maybe he just works with large enough quantities of yeast. I'm not sure.
If I eat enough yeast, can I self-produce it?
I don't think it works that way. I think there has to be the right conditions existing, right? So I think maybe you might have a genetic predisposition, or something in your body is not regulating correctly to kill all that yeast off. So if you chug a packet of yeast, no, I do not think you can induce this syndrome, all right? But if the right conditions persist in your body, it could happen, right? It's not a common disease, right? This does not happen regularly. It's kind of rare. Any other questions? Yeah?
Do people with the auto-brewery syndrome smell like alcohol?
Oh, I imagine it would probably smell in their breath. Yeah. Yeah. Google. Yeah, I don't know. All right, let's transition to our next case study. Who here has heard of the cyanide murders? All right, a few of you. If you haven't, that's okay too. If you have, great. I bet you haven't analyzed it in this way that we're doing unless it's specifically in the context of a biology classroom. I'm going to pass out some half sheets of questions. These sheets are yours to keep. You can write on them, do whatever you want with them. I am not collecting them. They are not being graded. It's simply just too much text for me to project up on the screen, so I'm giving you a handout. We are going to go through this part by part. And so what I will do is I will read out a part, right? There's 4 parts. And then I will give us some time to think about a couple of questions pertaining to that part. We'll talk about it, and then we'll move on to the next part. Right? So there's a lot of talking and discussion here. Let's see. I see they're all making their way around. Good. While things are being passed around, I'm going to give you some context for this case study. All right, so the context here is that we are going to imagine that we work at a medical examiner's office for a major metropolitan city. All right, as chief medical officer, you investigate suspicious deaths and provide toxicology services for the county. Unfortunately, it's been a busy week. Right? In the past 5 days, 7 people have died, all with similar symptoms. And it's my job, your job, to examine the data and determine the cause of death for these victims. All right, does everybody have the half sheet of questions? Perfect. Let's start with part 1. Let's learn how this all started. All right, so the first was a 12-year-old girl. Her parents said that she was awake in the middle of the night complaining of a stuffy nose and a sore throat. They gave her extra strength Tylenol and sent her back to bed. At 7 a.m. the next morning, the parents discovered that the girl had collapsed on the bathroom floor. An ambulance rushed the girl to a nearby hospital where she was pronounced dead. The same day, paramedics found the second victim unconscious on his kitchen floor after what they thought was an apparent heart attack. Sadly, the victim's brother and fiance also collapsed later that night while the family gathered to mourn his passing. Both had taken Tylenol to help them cope with their loss shortly before collapsing. Neither survived. In the next 4 days, 4 other similar deaths were reported, all in the same neighborhood, all with similar symptoms. And these symptoms included dizziness, confusion, headache, shortness of breath, vomiting, and most deaths were very rapid, occurring within a few hours of the onset of symptoms. All right, so there's a couple of questions on your handout that I want you to talk about in regards to this first part here.
More like and also the girl. And also considering like
So I don't think the next
that is something that is going
You can play the antagonists, like try to lead them astray.
They were all on the floor. Maybe there's something on the floor.
there's 2 guys.
Is there like a cyanide monster? Is that what it is?
Yeah.
It's like ghosts? Yeah. Oh, they're all together. And the 4 took Tylenol. I'll be working this 3 or 4.
Well, one of them this feels fun. This is like dimension.
Yeah, why don't we think about that?
Maybe they're in pain. Maybe they like they were vulnerable. I don't take Tylenol on Saturday.
You don't want to add what pain? Emotional pain.
Exactly. It is
Wait, Tylenol works for emotional pain?
What?
Tylenol works for emotional pain.
It does if you're addicted to it.
I did not know that.
I'm lying.
I'm a psych major. I did not know that can be.
It was the Tylenol, guys. It was a new, they came out with a new version of it.
And vomiting.
And rapid dosage.
7 individuals.
How do they die?
I think that's part 2. I think.
Oh, so they're running their chat?
No.
So they just died.
I know how to act now.
Why did you kill your daughter?
In reality, they're probably C's.
Well, I mean, think about it.
Well, cyanide.
It's a good idea to just take Tylenol for anything before you figure out what pain it is. Like, I don't know. If it's like, if there's like
It's pretty rad.
sneak out the ear, was it like don't want to have that one. What color is it?
All right, so you had a chance to think about these deaths.
It finds words.
All right, are there any similarities or connections between these 7 individuals that you saw? They all took Tylenol. That's a similarity. What else?
All the symptoms and how quickly they passed.
Yeah, all the symptoms were the same. Anything else?
They all live in the same area.
They're all in the same neighborhood. Yeah. So it's not like this is happening in Minnesota and in Texas and in New York, right? They're all in the same neighborhood. All right. So what questions would you ask the families? All right, you're a medical examiner. You want to figure out what's going on. You have no idea what's happening.
We were saying we would ask like when and where they bought the Tylenol.
When and where they bought the Tylenol. You had the same question.
Probably what did they eat and what did they do like in the 24 hours before this happened?
Yeah. What did they eat? What did they do? Right? Tylenol is a common denominator, but maybe it's just they all took Tylenol. It just happened by chance. What else could be causing it? What did they eat? What did they do? What else?
Maybe in the past, if they were exhibiting any like reactions to Tylenol or any sort of like over-the-counter medication.
Yeah, do they have a history of some sort of reaction with a medication? Maybe Tylenol specifically, maybe another type of medication. Yeah, these are all really great questions. Remember, we have no idea what's going on. We're trying to compile as much information as we possibly can. All right, so in your opinion, are these 7 deaths connected? Yes. And I'm giving you a case study. It's probably logical to assume that yes, they're all connected by something. All right, let's learn some more information here. So we're going to look at the autopsy report. So the autopsy report is completed, and after reading the file, you determined that the immediate cause of death was hypoxia, right, or suffocation or lack of oxygen. Tissue sections from the heart, lung, kidney, and liver all showed massive cell death. Staining with specific dyes showed major mitochondrial damage within the affected tissues. And the oxygen levels in the patient's blood was approximately 110 millimeters of mercury. That's the name of the unit there. Where the normal range is about 75 to 100. All right, take another few minutes here and answer the next 2 questions associated with part 2.
Cellular respiration.
Yeah.
We had our act out the task for us.
Yeah.
Do you have the act out?
Yeah.
Yeah.
I need it to show.
I think, well, which makes sense, cellular respiration is something that's interrupted.
Mitochondria and cellular respiration. It's a great connection. Why do we make that connection? That's what happens. So if we're affecting cellular respiration, could this lead to death? Getting energy. We need energy to live.
Their kidneys were squares.
Child immune system were slightly smaller.
Tylenol. Oh my god. Obviously without a brewery.
I feel like it's more ability of Tylenol combined with something else.
But I like that you noticed that.
It's that they can't use the oxygen in the blood.
Yes.
I mean, it's not there, they're still
Because
breathing.
I know there are some poisons that stuff.
Are you serious?
There's acid.
So like when it was all.
Are we noticing this
Well, it's like, yeah.
That there's more oxygen. Let me think for a second.
like extract or like actual?
Well, I'm guessing like the
Oh wait, then I don't know.
Okay, never been down to it.
I thought nobody.
You have to know what sign it was like.
I mean, I know about, I've known both of these cases. Have you never smelled it? It has very, it has a
If it closes up, it literally, I like to have like it feels like oxygen.
you have to eat a lot of
It doesn't feel like.
There's also cyanide.
Yes.
There's cyanide in animals.
There's something for me, like, it just doesn't feel like an external.
I don't know about that.
It's not usually.
You have to eat like, you have to blend like thousands of apples and then chug it. That's you make it. That's part of that.
Why is that?
I think it's only murder.
There's no way.
All right, so we learned some more information here. Right? We're beginning to understand the bigger picture a little bit better. So we're going to think about what we know about organelles, right? And specifically, the organelle that has showed a lot of damage, the mitochondria, right? What function in the cells do you think was interrupted because we had mitochondrial death? Yeah, cellular respiration. Right? Mitochondria are part of aerobic and anaerobic cellular respiration. Right? So something is interrupting this process. Now we're talking about humans here, so it's likely aerobic respiration. We don't have the ability for anaerobic respiration. Right? But if we saw the same thing in another animal that had that ability, yeah, definitely. Okay. Could the loss of functioning cellular respiration processes cause death? Yeah, why? What are we no longer making? Energy, ATP. Energy cannot be produced. ATP cannot be produced. That is a huge currency for the cells. Right? Cells use lots of ATP. If you don't have it, all these processes are going to start stopping, right? Which could lead to death. All right, what was the inconsistency that you might have picked up on in this report? What seemed unusual?
Higher level of oxygen in the blood, but lack of oxygen.
Yeah, so they actually had slightly elevated levels of oxygen in their blood, but they died from a lack of oxygen. So it doesn't seem to make sense, right? That's inconsistent. All right, so if you were a medical examiner, you'd pick up on that inconsistency and say, that seems odd. Let's investigate this further, which is exactly what we're going to do. We're going to get a detailed analysis. All right, so detailed analysis of the damaged cells showed that ATP levels in the mitochondria were very low. Levels of pyruvate and acetyl coenzyme A were normal. You begin to suspect a malfunction of a specific cellular metabolic pathway. And so you request a more detailed analysis of the subcellular components of the affected cells from the autopsy. The levels of key metabolites are listed below. All right, so I want you to look at these metabolites. Think about, are they normal? Are they abnormal? Where are they used in the process of cellular respiration? And ultimately, what stage of cellular respiration do you hypothesize is being affected by these deaths? What is causing these deaths? I'll give you 5 minutes because there's a bit more to this question.
I will assume
Obviously.
Yeah, the products are actually
First one looks normal. Second one also looks nice. Oh my word, NAD plus is not normal.
Not normal.
NADH is normal.
Yeah, but like which side?
What? Do you read Chinese?
No, I'm not Chinese. So
No,
true.
I mean I know, but I'm surprised.
Well, I know that's Chinese.
I'm just
because
gonna
it
put
is
this
Chinese.
Good point.
A lot of Asians speak music.
Yeah, it is.
I remember it.
Let me think.
It sounds like friends.
The first thing I would think of is that there's a problem with young.
At some point, I will file up.
What's that? What's that? Everyone knows what right? That like changes yeah, that is putting
So much cyanide.
I'm mixing these up, I know it. There's those type of terminology. I'm not really sure.
I would talk about this. I want to be a missing 411 case. It's so bad. I want to go missing under investigation.
Also, it feels like
And found in a hospital distance away. So much fun. Or like I want to get into this and like
roll into
be trapped in the word. And then hide in This is fact. No, the main character wins. But I will be like, you guys have to. I think it'd be fun. And I'm like, but the only way you can do it is if you guys take
We regenerate NAD. Which of them did we
You have to,
say
like,
we had to? Oh yeah, we didn't want to build up
make a hat.
We wanted like more NAD plus so that we can make.
How about you? How would you kick him?
Which I think is good with the house on top of electron temperature.
Or you should really move to Florida. That's where it's gonna happen.
Okay, so if it continues,
That's like weirdly
what do I think? It's somewhat happens there.
If you had
Let me search the agents. I'm getting
the way they would get rid of it is like it's like at the counter where like took out your whole like it's not like see anything. It's kind of like this thing. Like they just wanted to do that, to be honest.
That's why I like that. Yeah. It's never mind.
Like different species.
Yeah, no, that's not what I'm looking for. I'm looking for the other way around. There's a buildup of NAH. Or it was like back into NAD plus, so it is the electron transport chain. They do get another electron. Wait, no, we found it the other NAD donates its electrons, uses the hydrogen, and
like
then
that guy who got
turns to NAD plus. Yeah.
But like I'd like to be dead or have no Yeah, I don't understand.
Well, you just said you wanted.
I see it very
Exactly.
so excited.
Or like, if someone could like hollow out one of the stuff me in there. I just know it's just
They are
but that's
well,
how we found.
they are substrates and they are products because I don't. In the electron transport chain, NADH is a substrate, NAD plus is a product. So,
see how the breakdown.
So yeah, so then we have a buildup of NADH because we can't,
What stage electrons?
because we can't
I want to find an electron.
make that into NAD plus because the electron transfer
Oxygen is not finalized here. Yeah,
because there's like the cell isn't able to use oxygen.
oxygen takes page from NAD.
Yeah, but it's not the same.
Which is why.
All right, let's talk about this detailed analysis. So we have glucose listed. Where is glucose listed? What stage is glucose used in cellular respiration? In glycolysis, the first stage, right? That's where glucose is starting to be broken down. All right, and the levels for glucose look pretty normal. All right, our next metabolite is pyruvate. Where is pyruvate produced? What stage? Stage 1 still. Yep, still glycolysis. It's being produced in glycolysis. What stage is pyruvate being broken down in?
Krebs cycle.
Stage 2, breakdown of pyruvate. Yep. All right, and pyruvate, that looks pretty normal. So does that indicate that glycolysis is still working? Yeah, cells are still going through glycolysis. Okay. Breakdown of pyruvate, and then we have these other molecules, NADH and NAD+. Let's look at NADH. Right? Where is NADH produced in the largest quantities? In what stage of cellular respiration?
Electron transport chain.
Not quite. It doesn't play a role in the electron transport chain, but the Krebs cycle, right? The citric acid cycle. Right? That is what's producing the largest amounts of NADH. Right? Now we know NADH can also be produced in glycolysis, right? But not as large of a quantity as the citric acid cycle. And what do we notice about NADH in our analysis here? There's an excess. All right, so that indicates that it's still being made, right? Because there's a lot of it, right? So if it's still being made, what cycle is still working? The citric acid cycle is still working, all right? But it's not being used up, it's kind of accumulating. There's an excess of it. What stage uses the NADH?
Electron transport chain.
Yeah, the fourth stage, the electron transport chain and oxidative phosphorylation, right? So if oxidative phosphorylation uses NADH, does that mean that fourth stage is happening? No. All right, so now we've narrowed it down, right? So we've gone from knowing that it's the Tylenol to knowing that it's affecting cellular respiration to knowing specifically what stage in cellular respiration is being affected. All right, we can get more detailed. So now we want to know, how is this being affected? Right? Why is NADH not being used? Which leads us to this last part here. All right, so we are now convinced that you know the cause of death for these victims and quickly report back to the police, as this is a very dangerous situation. After realizing that the electron transport chain was no longer functioning, you start to suspect poisoning and ran a blood test for various poisons that you knew affected the electron transport chain. The test of all 7 patients came back positive for cyanide. Cyanide irreversibly binds to cytochrome C oxidase of the electron transport chain and prevents the transfer of electrons to oxygen, the final electron acceptor. All right, so cytochrome C oxidase is this purple molecule in the diagram here, right? It is an enzyme. Something has irreversibly bound to this enzyme, right? So thinking about irreversible inhibitors here. It no longer functions, which means it can no longer pass an electron onto an oxygen to complete that electron transport chain. I want you to work through the last 4 questions to figure out why is cyanide, like, why did this patient die, basically, from lack of oxygen when they had oxygen? What is cyanide's role in this?
The mitochondria is dead and they can't
block the production because now it would be prevented out of the country. Just like block the stuff right before oxygen. Even if they took it. Which is also what there's other changes.
The cops. The cops.
Oh, because it's bonded. Yeah.
Sorry, there's still no wheel there.
Now.
Yeah, I do.
Because they weren't able artificial respiration, but not able to use oxygen. That's why they added zero.
What if they got?
If you, they weren't breathing properly, they just couldn't use it. So like, I'm gonna put a respirator on you, and now they just have more oxygen.
And NaH. Yeah, it was like the step right before that.
What can we do to save? Is there anything we can do?
No. No. Also, like, I would be really upset.
Don't tell me it's not no, because
Yes, it's universal bond.
I was also already passed because I
If you give them more oxygen, they're still not.
Yeah, I'm not
Somebody poisoned the Tylenol.
oh really?
Yeah, this is real.
So what's the source of it?
All right.
It was the Tylenol. Some of the Tylenol. Some of the Tylenol replaced guy who lived in the neighborhood, I guess. Just chucked it in for some
Looking at fermentation. So we're thinking about this question. Have you slightly different process?
There weren't really many
It's losing hydrogen. That's why we have half.
Yeah.
Yep. And when it gains electrons, when it gains the hydrogen, then it goes back.
Also, like, if you open it gets that keeps it safe from the ship from the carrier.
So you carry these electrons
Not saying I'm listening.
NADH, you probably lose that level, like this can't be bad.
My sister just died and I
So we want to make sure it's constantly cycling. Maybe they were, I think every right now.
Triplets, just two siblings.
All part of that.
It's not related. That was just a random Yeah, these are three separate places. Other houses. Hey, Steven, did you know that like 8 of the houses just should we like talk about it? Someone was killed in my old house right before we got it.
I remember that, but
I think so. Well, I mean, it wasn't really a murder. He like smoked that with the color of the world.
they are all of that together.
Anyway, it was his choice. Huh? If ghosts are real,
But they're more for the purpose of jogging my memory, like practice.
where have
No,
I don't know. I listened to it.
let me show you this. This is the thing with which I used to take those citations for. This is learning techniques. I am writing this down. I'm writing out.
It's safer for you.
I know, it's really short. Now, I label them different constants because they are, this is thought identity.
You should look it up.
It's like, I just have these ones in here to tell me where I generated these ideas from.
I didn't know that.
Oh, yeah. Because you can find more. I don't know, where did you go? It's just, I was yes, elementary school for 12 years in this region. It was called
9 days.
oh, yeah.
Yeah. They actually didn't do any tests.
You know, I guess the
They just paid off like one of
But it's like,
Do you teach them when you were a tutor, you taught like Chinese? Yeah. Not German.
Alright, let's check out our case study here. So, what effect would cyanide have on the electron transport chain and the production of ATP? Would the production of ATP continue? No. Why not? Yeah, it affects the electrochemical gradient, right? How does it affect the electrochemical gradient?
ATP is made through the oxidation of NAD plus or NADH. So when it's not getting rid of that hydrogen, it's not making ATP.
Yeah, we're not building up that hydrogen gradient that makes ATP synthase work to make ATP. Yeah. Let's see here. So, oxygen. Oxygen is not being used. Right, what does that mean about the electron transport chain? Is the electron transport chain working? No, it can work, but it's backed up, right? If I were to think of an analogy here, I would think about this cytochrome oxidase as kind of being like a doctor's office, and you're waiting in line maybe for like a flu shot or something, right? And the person in the doctor's office is never going to leave, right? That means you're never going to move up in line, right? Kind of the same thing too. We have all these electrons waiting in line in the electron transport chain. They're never going to get to this cytochrome oxidase, right? So it's all backed up, which means we can no longer donate any more electrons from the NADH, which means we have high levels of NADH, right? They can't give away their electrons because the electron transport chain is at capacity, because the electron in the cytochrome oxidase cannot be given away to that oxygen. It's never going to leave the doctor's office, essentially, right? So these patients, they died from lack of oxygen, but it might be more accurate to say that they died from lack of being able to use the oxygen, right? So if we gave them artificial respiration, would that have saved these patients? No, right? Because they already had access to oxygen, right? Their bodies simply could not utilize it in the electron transport chain. All right, let's see. So what is the possible source of cyanide poisoning here? The Tylenol bottles. Yeah. So where the cyanide poisoning entered into their system was the Tylenol. Remember, all the way back to part 1, that common denominator was that they all took Tylenol, and they all took Tylenol and lived in the same area. So likely they all bought it from the same store or a couple of stores, right? So Tylenol was the culprit here. So how should public officials respond to this poisoning? What should we do in response here? Investigate the pharmacies.
Recall all of the Tylenol.
Recall the Tylenol. What else?
Add tamper-proof packaging.
Add tamper-proof packaging? Yes, all of these happened. All right, so this was a real case. This happened in Chicago in the early 80s. Right, some individual or individuals, we don't know because the suspect was never caught, actually took some Tylenol, emptied out the contents that made it Tylenol, and filled it back up with cyanide. So it looked like a Tylenol pill, but it had cyanide on the inside. and put it randomly into bottles on shelves of a store, right? So these are really random killings. These are not targeted. Whoever took that Tylenol would die, right? And so the Tylenol was the case here. And that's how it entered into these victims' homes. And it all happened in a relatively contained area, this neighborhood in Chicago. Now the result of which, right, after we discovered it was the Tylenol, yes, they investigated the companies that made the Tylenol, right? They investigated, did the cyanide get put into the tablets at the plant that made the Tylenol? Maybe they were all in the same box, they were all shipped to the same store. They investigated individuals that they thought could be connected to this case based on various leads, but they never had enough concrete evidence to convict anybody. So we still don't know who really did it. But the good thing that came out of this is now we have tamper-proof bottles, right? Before this, before the Tylenol poisonings, we did not have this tamper-proof cap, right? You could unscrew a cap, slip in a couple extra Tylenol that had cyanide in there, close it back up. No one would be none the wiser, right? But Tylenol, right, they're a big company. They don't want people to not buy Tylenol. They have to save face. So they came up with this protective measure to put on this tamper-proof packaging to gain back the public's trust, right? Not only this tamper-proof packaging on the bottle itself, but now the bottles were also packaged in boxes. You also have to open the box and then open the tamper-proof packaging to slip in a poison pill, right? So that did gain the trust of the public back, right? And there was actually some cases afterwards where cyanide poisoning popped up again. It's very strange. I encourage you to watch the documentaries. There's some on Netflix, there's some on YouTube. BuzzFeed Unsolved has a good one here. But it's still unsolved. We don't know who did it. And it's a very, very sad case. All right. Yeah.
Would the tamper-proof bottle prevent like further poisoning if it was done by someone in the factory?
No. Yeah, so it could still, you could still get poison into Tylenol, right, in a factory. It's not an end-all solution. You could also say somebody could buy this and open it up and make it look like it was never tampered with and put it back on the shelf, right? But hopefully there's not enough individuals in the world that are going to do that, right? Have some faith in humanity. But yeah. Any other questions related to this case? All right, so hopefully you saw how your knowledge of cellular respiration helps you apply it to a real world scenario here. All right, so I love scenario-based questions. They make you think and put into some problem solving into your answers here. So you would expect on an exam, I might ask questions similar to this. Something has happened. What is going on? Why is this the case? All right. We're going to lead into our assignment, right? I don't like leaving assignments open-ended. So let's talk about the scientific literacy assignment I had to do on misleading statistics. And so I want you in the next 4 minutes to look at the next 3 graphs and tell me why these graphs presented could be misleading. And they're all misleading for different reasons. I have given each of them their own slide if you want to look at them kind of blown up a little bit, right? But I tried to incorporate sugar from the Nobel, Tylenol that we talked about the cyanide murders. So give these a look and then we'll talk about them.
Percentage of calories from added sugar. Oh, no, it's reality.
That one doesn't explain what percent of calories from doesn't fully explain it.
It's also not that big of a difference.
And that one's scale is really zoomed in.
It looks like it's dropping, but it's percentage. I mean, they didn't say how much there is.
It's a little skin, right?
Also,
Yeah, so like
Exactly. So it's like So they have like 100 grams of sugar, and then if the whole thing is like 200 grams, and then you have like in there 10 grams of sugar, that's
It's really not that bad. It's really not.
Oh,
That makes sense.
never mind. I take it back.
Hey, my mom fixed There was cyanide.
I think it's trying to say that, like,
Yes, the baby died.
Could you, yeah, it's comparing rate.
The baby's already got the cyanide. We know, now that we know the baby is well, I was a little worried you were gonna go, no, it's more worth it. Honestly. I mean, that is so
Well, yeah, also like.
but it looks used compared to that.
Two potential. 2 plus. I don't know.
Yeah. It's not really a big deal. I just want
like I don't know. Go back to
Yeah, it does look very different.
so. Oh my word! If you look into the history of psychology, you see the most insane part of human history.
I like that one.
I want to go into neuroscience.
Wild you can be like out there and some
So yeah, I've been building the best skill set though.
That's my thinking. No, I'm thinking they did. I'm thinking they all did.
I mean, it could be a mixture of both. There maybe it was intentional, maybe it was unintentional.
What did I mean by That's what I thought.
Like totally. That they ate food per day, food percentage, like. Yeah, yeah.
If I had to choose an answer, yeah.
Favorite mountain? I like downhill skis, so I'm gonna say the Rockies. Yes. I love skiing. I've never been to the Appalachians, but
But I know it's crazy. It's so
I really like
Yes, I love that explanation. I know it's great.
thing.
This is supposed to be the mountains in California. You get set on fire there.
Sierra Nevadas out there. I love those too.
It's never good.
I did a lot of my thesis research.
Like a lot of times they conclude that we're
It's expensive. It's really
Your house is going to
well, that's what women are also
or like, or you're gonna have your house is gonna work apart.
Well, natural disasters. The person that I work with
about the
is
that is the major
All right, let's look at these graphs here and talk about why they could be misleading. All right, let's start with the easy one. Let's start with gun deaths in Florida. Why is this graph misleading?
You would expect like the y-axis to start with 0, but it actually starts from 1,000, and most people, when they're doing a manual calculator, it's skipping the graph.
Yeah.
Actually playing the opposite.
Yeah. So at first glance, it looks like after this law was enacted, gun deaths dramatically dropped. But if you take a few more seconds to look at the graph, you would notice that the axes are kind of flipped, right? It's going from a descending order rather than ascending. right? So it might be more accurate to make it look like this, right? So where the zero murders committed using firearms is typically where you would expect the 0 to be on a graph in the bottom corner, right? So it looks like after this law was enacted, maybe there was an initial spike. Now, I don't know anything about this law, and I'm not putting into the politics of it here, right? But this graph is just it's so misleading that it might have been done intentionally, right? This is a very weird way to present data. I don't think anyone would intentionally present data this way unless they were trying to misconstrue a message. Who knows? I don't know anything about the law or how it unfolded in real time here. Anything else about this graph that anyone wanted to mention?
The 2010s are really condensed compared to the 90s and early 2000s.
I didn't even notice that, yeah. If you look at the scale, right, the 1900s, or 1990s, 2000s, and 2010s, right, the 2010s are really condensed. And maybe they only had data to like 2014, well, yep, till 2014, but it makes it look like it's the entire decade. It's a little misleading. All right, let's look at another one here, thinking about our Nobel conference. I have this visual, it's a table here, showing the percent of calories from added sugars. What could be misleading about this visual?
The source of this graph is from the website that wants to eat more sugar.
Yeah. I should link the source in the notes here, but it's from a website that wants you to eat sugar. Saying sugar's not that bad for you, it's not demonizing sugar.
It also sort of like leaves out like the increased prevalence of natural sweeteners instead of sugar over time.
Yeah, so it leaves out some information like the natural sweeteners or artificial sweeteners.
I just think there's like no context in general. Like looking at it, like I have no idea what's really like what's even going on.
Yeah, context is missing.
It doesn't explain what exactly they're measuring the percent of.
Yeah. Like percent of what? Percent of your total diet, percent of the foods that we measured, percent of what you're actually eating.
They don't mention like it's a percentage. It's not an actual amount. So if you eat like a huge donut, you're still versus like a chocolate bar, like you're still injecting the same amount of sugar, but like the different percentage.
Yeah, so looking at it at percentage instead of like a finite number. Yeah. There's another piece here too that maybe if you watch the Nobel conference of certain speakers, you might have picked up on. What else is missing from this? Could be portion
That's
sizes.
another thing, so like percent of calories, total calories will increase anyways, so like reduce them.
Mhm. What about how long has sugar been in our diet? Not very long, but longer than 2000. Right? So actually, this is a good example of like cherry picking some data. So if I were to look at this graph, this is a graph, it's comparing it to diabetes here, but what I want you to notice is the amount of sugar consumed over time. It starts in the 1950s and ends around, 2020 here, right? And we can see there's a general increase in the amount of sugar in our diet over time. But around the 2000s, it starts to decrease a little bit, right? But the amount in our diet, even after this decrease, is still way more than in the 1950s, right? So they kind of cherry-picked some information. They only gave us information since the decline in 1999. They're not saying that it's still a lot higher than our diets were in the 50s, right? We're still consuming a lot of sugar, right? So yes, this did come from a source that was trying to say sugar is, I don't know if it said it was good for us, but they are promoting sugar. Any questions, comments about this? All right. My next one is kind of a hot button issue at the moment, right? This is a graph showing autism rates among children by age 8, right? What could be misleading about this graph?
Historical context of how and how much we've been testing children, how we diagnose.
We're missing context about how diagnoses are happening and how many, like, how prevalent is it for someone to go get diagnosed, right? Nowadays, there's less stigma associated with getting diagnosed. We are increasing awareness, right? What else is misleading or hard about this graph?
The x-axis goes from 2000 to 1992, 2004, 1996. It's not in any order.
It's confusing. Yes. So it's surveillance year and their birth year. So they surveilled these individuals in 2000, but they were born in 1992. It's confusing, right? You really have to look at it to understand it. It's not obvious, right? So it's confusing in how they made the graph. What else? Is this data inaccurate? This data is actually not inaccurate. If we look at the data itself, the data is actually true. There has been a rise in autism rates in children by age 8, right? And the data shows that. This is a graph from Nature showing a rise in both males and females. The data is accurate, but it's all about context, right? It's accurate, but it's because we've had an increase in awareness. There's been an increase in individuals seeking the diagnosis.
It's also not taking into account population growth and the fact that autism was relatively recent disorder that like we.
Yeah, population growth, and that it's a relatively recent disorder that's been described a little bit more in depth now. So you can get like an autistic diagnosis that's it's along a spectrum. It's not like you're autistic or not. It's a spectrum. So there's a rise of people on the spectrum.
I think it is important to, like, you even showed it like the boys versus girls. Like, we don't know what population you're taking. Are you taking more girls and it's gonna be less because you're taking that, or is it gonna be more, like, based off that?
I think that gets kind of into the, I don't know how I would describe it, like the psychology or ethics or like just the culture of autism, that sometimes boys are diagnosed more often than girls for a variety of reasons. There's a whole topic we could go on there. But in this case, the data is accurate, the context is missing, and we're trying to compare it to the use of Tylenol that pregnant women are taking, but there's no data about the Tylenol on this graph, right? So we've been hearing this in the headlines, Tylenol can cause autism if pregnant women are taking it, but we're not comparing it against anything, and that's the context by which this graph is being given, right? So this is a little bit of a trickier one, right? The graph is accurate, but it's all about context. It's how they're using the graph. Any final thoughts on these graphs, on the assignment? And if I were to ask you, if you're looking at a graph, what are you going to pay close attention to going forward to know that the graph is not misleading? Yeah, who is making it? Who's publishing it? What might their motives be? What else?
Is relevant data out?
Yeah, what data is included or excluded?
Look at whether or not they have figure captions.
Figure captions, yes, figure caption gives us a lot of context behind what we are looking at. Figure captions are great as we're all learning in the lab. Anything else?
Just the general structure of the graph.
Yeah, general structure, we can look at scale, are they zooming in to make small differences, look at large differences? Yeah. All right, so as you continue forward in your science careers or science adjacent careers, pay attention to these graphs, right? This doesn't just happen in science, it happens in every discipline that you could imagine. Graphs can always be misleading. All right. My last class was way more animated about this topic, and so we spent a lot of time talking about these graphs. I'm going to do a very abrupt change of topic now. We're going to talk about photosynthesis. All right, So that video I had you watch talked about photosynthesis, but more specifically about the structure of a leaf and where photosynthesis is occurring. It was a very, very broad introduction to photosynthesis. Now, my last class, we did not get through very many of these slides. We'll see how many slides we get through, and it's likely that I'll let you go early because I don't want our classes to be too out of sync here. All right, well, let's start with a review. All right, let's think about the structures of a chloroplast. I want you to, for all of those labels except for number 6, because we didn't talk about number 6, tell me what those structures are labeling, and then tell me where would I find chlorophyll molecules, or the pigments.
What are the types called? I don't even know.
Yeah? Of correlation and causation?
To be fair, it's been 2 years since I took the last psychology course on this.
Yeah. Oh, let's see.
I mean, do you mind if I ask my psych professor for one?
You could, honestly, you could type into Google, what's the case of correlation does not equal causation, and I'd be fine with that too.
Splendid.
Yeah.
I'm surprised that you didn't talk about the beige.
Where's the
I talked about the Simpson paradox. I'm surprised you didn't talk about the Bayesian thing, because that's the one where, like, you have a 99% accurate test, but then you're only like the disease. You're only
fluid between
how common it is. It's only like 1 out of 10,000. And then so even if you get tested positive for this disease, you are not likely to actually have the disease.
That's true. That is something that maybe we could be talking about here.
Most ridiculous, like, fun things I've been learning. And
Yeah. Yeah.
I listened to this lecture with my mom on statistics.
Okay.
That is where statistics is like, a lot of people think science is super objective, but I mean, it's interpreted by humans. It's all.
Yeah, there's always bias. We just can't see it. Whether innate, external, known, there's always bias.
Yeah, a lot of people who are in the science field, they think that, like, if science didn't, if it's not supported by science, it's not true. Not necessarily. It just, like, I mean, in terms of, for example, is there a God? Like, current technology just cannot detect that. But if God is a fourth or fifth dimensional being, then that explains it. That's strange.
Right, there's limits to science. Yeah.
Of course.
I agree. I like to think of it in terms of a lot of times we think like traditional medicines don't work. Yeah. But it's still an untraditional medicine. It's harder to produce than we see something else.
That and also the fact that, like, our technologies, like, what are the intents, what metabolic, if there's like subtle metabolic changes that's happening as we progress.
Yeah. Like we know they work to some extent because they've been used for such a long time. Are they as effective as the outdated medication? Maybe not. But is there some effect?
In terms of specifically questions I've been used, like acupuncture for a while, they will do some of what you get it right. They do better. Synthetic memory. Like during COVID, I used this method that got me resting in like 4 hours. Yeah, I know. I use that because how medicine did not work.
Okay. Because there's something to it. Yeah. All right, let's look at the structure of a chloroplast here. Let's start with the easiest one in my opinion. What is number 5? Thylakoids. Yes, number 5 is the thylakoid. I like to think of them as stacks of pancakes. Right? They look like flattened stacks of pancakes. If we stack many thylakoids together, does anyone know the name for like a stack of these pancakes? It's the gram. It's not on this list, right? But it's a gram. Stack of thylakoids is a gram. All right. Let's take a look at number 3 here, kind of this area that's in this darker green. What would you label that as? I think I heard it. Outer membrane, yeah, I would say this is an outer membrane, right? And then if we look at number one, what would I label number one as?
Inner membrane.
The inner membrane, right? Because we have two membranes. We're thinking back to the theory of endosymbiosis as to why it has two membranes here. Number two then, right, I would say is the intermembrane space. It's just the space between. All right, so then what is number four? Yeah, it's a space surrounding the thylakoids inside of the chloroplast. It is the stroma, right? So that is the space surrounding the thylakoids. Does anybody know the name for the space inside of the thylakoids? The lumen, yes. So when we continue our talk about photosynthesis, you'll often see stroma and luma, because we're going to be talking about membranes. On one side is the stroma, on one side is the lumen. All right, where would we find the chlorophyll molecules in a chloroplast? In the thylakoid membranes. Yes, they're going to be embedded within the thylakoid membrane. And we will see that in the next few slides that we get here. All right. Let's start thinking about photosynthesis. And we're going to break it down. So let's just look at the broad overview of photosynthesis. Photosynthesis has two stages. It has the light reactions, and it has the Kelvin cycle, often called the dark reactions. Right? I use the Kelvin cycle instead of the dark reactions, right? Because the Kelvin cycle can happen when there is light. It doesn't have to happen in the dark, right? But it doesn't need light. So the light reactions use light energy from the sun. Right? We are taking in sunlight. And in the process of taking in sunlight, we are making ATP. We are making NADPH. Right? Notice that sounds like NADH. You can think of it as like the plant version of NADH. It's NADPH. It's still an electron carrier. Does the same function. And we're also producing some oxygen. Right? So the light reactions, using light, producing ATP and NADPH. The Kelvin cycle then uses that ATP and NADPH produced by the light reactions to make carbohydrates. Right? So they'll take in carbon dioxide from the air, right, and incorporate those molecules of carbon dioxide and turn it into glucose. All right, so these reactions kind of work in tandem. Right, so the light reactions produce ATP and NADPH, which are used in the Kelvin cycle. Right, once those ATP and NADPH are used, right, they become ADP and NAD plus, BP plus, similar to how we saw in like fermentation and cellular respiration, and they'll be regenerated in the light reactions and continue on in the cycle. So they work in tandem. All right. Let's start by talking about the light reactions. And in order to talk about the light reactions, we have to understand light itself. So what is light energy? All right, so light is a type of electromagnetic radiation. Right? It travels in waves. And these waves can vary in their wavelengths. They can be really short wavelengths. They can be really long wavelengths. And we kind of see that on this graph here. Towards the side where it's more blue here, wavelengths tend to be a little bit shorter, a little bit closer together. The opposite end of the spectrum, they tend to be spaced out a little bit more. Now this is important because depending on the wavelength, it will determine how much energy is carried in the light. All right, we'll come back to that. But light can also behave as a particle. And that's what we're really going to focus on, is this light behaving as a particle, and those particles are called photons. Now, different than protons, right? Protons, they're in atoms. Photons are discrete units of light. And I like to keep it, remember it as photo, right? I have to use light to look at photo, so photons. They're discrete packages that contain energy. And the amount of energy they contain depends on the wavelength of light. Okay, and again, we're going to come back to that. It's all going to fit into the bigger picture here. All right, so we have these discrete packages. We have these photons. What happens to these photons? All right, well, when a photon strikes a surface, three things can happen. The photon can either be absorbed, it can be transmitted, or it can be reflected. All right, now in a plant, all three are going to happen, but we're going to focus on when it's absorbed. All right, so let's look at all three. Light is coming into this chloroplast. Right? It's interacting with our thylakoids. Light can be reflected back, right? So it bounces off of the thylakoids, reflects back at you. If it's being reflected back, that light is not being used. The light can simply travel through the thylakoids and be transmitted. Right? It's just traveling through. Again, it's not being used. It's just being transmitted through. Or the light can be absorbed. And when the light is absorbed, that's when we're actually harnessing the energy contained in these photons. All right, so light can be absorbed by the chloroplasts, and specifically, they are absorbed by pigments. Pigments that are embedded within the thylakoids here. We can see the most common pigment actually looks like this one down below, are chlorophyll pigments. Right? Now, if we look at, say, outside right now, there's still a lot of green, which is kind of unusual for October. I would expect maybe some more changing of the leaves at this point. But there's a lot of green out there. When you look at a plant, you think green. We see the green because plants cannot use green light. That green light is part of the light that's being reflected back towards us. Right? So we're seeing green because green is not being used. All right, this is my last slide before we are done here. All right, so these photons, they're being absorbed. Okay. What happens when this energy is captured, when it's absorbed? All right, well, if we look at what happens, let's take a look at this electron. Right, this electron, where a photon hits it, this electron becomes excited. Right? It gains some energy. It enters into what we would call the excited state. And if we see this electron, it's actually moving further away from the nucleus. It's actually jumping to another shell of the atom. Right? It's moving further away. It's being really excited. Now this is unstable. Right? Having an electron that is this excited, that has this much energy, is unstable. It cannot stay this way. It has to lose that energy. So that energy is lost in a variety of ways, and all three happen in a plant. Right? But only one is really associated with photosynthesis. The electron can release the energy by means of heat. Right? It gives off some heat. The electron comes back down to its ground state, its stable state. Right? Like heat radiating off of a laptop here. The electron could also emit light. Right? It could fluoresce. So like bioluminescence. Right? That is light being given off by organisms. Right? That's how it can lose its energy, go back to its ground state. The third way, and this is the way that photosynthesis works, is that this light, these excited electrons from that light, can actually be transferred to another molecule, can be captured by another molecule. And it's the capturing of this electron by another molecule that enables photosynthesis to happen. All right. Next Tuesday, we're going to go over actually the steps of photosynthesis, and it will all be on the exam. What I don't think will be on the exam is the variations of photosynthesis. I don't think we're going to get to the variations, right? But there's a lot of context. I'm going to try to get these slides up ahead of time so you can take a look at it and start studying ahead.
Oh, shoot. Do you like my house invitation that I made? I didn't have a pencil, so I just had to do it. Not content. That's pretty good for not. Oh, these two assignments all for today. I know they're not due today. I was like so confused.
On Moodle, said they were due. I just changed that like an hour ago.
Okay, I
Yeah.
see. 'Cause that was so confused. I got told
not
Yes,
in it.
refresh the page. I changed
Got
it
it.
right before I
I just
started.
looked this morning and I was like, wait a minute.
Yes.
I got so fooled by the calendar.
Yeah, I had to rearrange the schedule a little bit. Yeah.
I was like, this isn't covered in class. I'm like, what the heck? Do I have to pull it from knowledge from
No,
years ago?
no.
I forgot most of the terminologies from that.
Okay. Oh, yeah.
Did you move like
Yes.
what I do with it, like do it online or something?
Yes. Email me that you'll be going. I will send you that slide that we do in class so that you can see what's on it.
It's the least physical practice.
I love doing case studies. They just take up so much class time that I can't do as many. Yes, they're a lot of fun.
It was harder.
Oh, it was?